you will leave today feeling like you absolutely could do. In today's world, life sciences research is no longer confined to glassware and white coats. It is about understanding why you are doing something, knowing how much technique fits into bigger picture, and feeling confident enough to discuss, apply, or even teach these techniques to others. So whether you are aiming to publish your first paper, land your dream research internship, or simply make sense of what is happening in the lab, this session is designed with you in mind. For those joining us for the first time, I am Ali Daras, a proud student of genetics and molecular biology at the University of Health Sciences, and someone who is fallen in love with the process of scientific discovery. Over the past few years, I have had the opportunity to work in areas ranging from bioinformatics to stem cell therapy, the molecular diagnostics, the genetics and evolution. And trust me, the tools we are discussing today have been at the heart of every breakthrough I have experienced. Today, we will walk through the practical side of research from sample collection and nucleic acid extraction to PCR to primer design and even checking the DNA quality. Uses tools like NanoDrop and gel electrophoresis. The goal is simple: no fear, no fluff, just crystal clear understanding. So grab your notebooks, open your minds, and get ready because we are about to take a deep dive into the hands-on workflow of molecular biology and life sciences in research, starting right from the first crucial step. So let's begin. Now I am going to share the screen to all of you. Do you see the screen? Kindly tell me in the chat box. Do you see the screen? Okay, thank you. Okay, this is the day two of our workshop, which is about the theme of our workshop is life sciences research workflow from hypothesis to publication. It is day two, learn the techniques and tools without fear. It is the main agenda of our day two session. So let's begin. Okay, now let's start the second day session by my introduction. My name is Adit Ross. As already I said, I am PhD genetics and molecular biology scholar at the University of Health Sciences and have a good brings a wealth of knowledge and hands-on experience in bioinformatics, cell therapy, genomics, genetics and evolution, molecular diagnostics. So, what is the session overview? The day two is all about understanding the lab tools and techniques without being overwhelmed by technical issues. The goal is to introduce each concept in simple language so students or the researchers can confidently discuss and apply it even if they have never stepped into a lab. So, What is the workflow of the life sciences research? So first of all, we will talk about the sample collection. Then we will talk about the nucleic acid extraction, in which we'll talk about the DNA extraction. Then we will talk about the RNA extraction, and after the DNA extraction and RNA extraction, we will dive into the NanoDrop. It is about the quantification. It is about the quantification of extracted sample or we got the product. And also we will also discuss about the gel electrophoresis. Why? Because in NanoDrop, the disadvantage of NanoDrop is the degraded. Kindly keep in mind, the degraded DNA is not seen in NanoDrop. If the DNA is degraded, the quantity of the DNA is seen in NanoDrop. But in gel electrophoresis, we can also see the degraded DNA. So it is old but have advantage to see the degraded DNA in gel electrophoresis. And after it, we will also discuss the PCR, the polymerase chain reaction, and then we'll talk about the primer designing, and then the bioinformatics tools, and at the end, we will also discuss the mini task, and we'll give the mini tasks of day two. So start from the techniques and tools the tools used in life sciences labs. So first of all, we use the most important and mostly used equipments is micropipers. The three types of micropipers used in uh, life sciences research lab. So first of all, for example, it is P10. For example, it is P10. Then it is like P20, and this is like P1000. Okay, this is just example. So for we use the tips at the end of it. The tip used for P10 or the less than P10 is white. And for P20 we use yellow tips. And for P1000 we use the blue tips. And we also use the swabs for collecting the samples. We also use different types of containers. We also use thermocycler. It is actually the thermocycler we use for PCR, the polymerase chain reaction. We will talk about it later. We also use the centrifuge. Also use the centrifuge machine for separation. We also use the reagents in molecular biology labs, and we also use the the apple drops. So this is gel electrophoresis, in which this is actually the container. This is the power supply, and this is gel. We made this,
Sorry,
and
please, are you sharing your screen?
sorry.
Okay, I've seen it now.
So the other contaminations means there may be a salt contaminations or like that. And the next ratio we got is A 260 by 230. Okay, but what it indicates, if it is above the 2.0, it indicates no salt contamination. And the quantification by NanoDrop, this type of screen we will got, okay, and this type of graph, this this peak tells up about the DNA and RNA, and this is about the protein, and this is about the salt, like that. That's 260 by 230. It is about salt contamination, and 260 by 280 is about protein contamination. So the 260 by 280, it must be 1.8 to 2.0, and 260 by 230 must be 2.0. Must be greater than 2.0. It may be 2.1, it may be 2.2, maybe 2.3. It is the better value. But if it is 1.7 like that, there is the definitely salt contamination. So now we'll talk about the gel electrophoresis. So a method used to separate a mixed population of DNA or RNA based and sizes and charge. Okay, there is a mistake. It is based on a method used to separate mixed population of DNA or RNA based on sizes and charge. Okay, DNA migrates through a highly cross-linked agarose matrix. It is very closely linked. Okay, it is like that, and DNA is passed from that. Gel is composed of agarose. It is like gelatin and pH buffer for maintaining the pH. It may be a TAE. It also is TB. We also use the etherium bromide, which is used to stain the DNA bands. It is actually used to visualize the DNA. Why? Because as we know, like that, this is DNA. Okay, and the one is major groove, and the second one is minor groove. Okay, so the etherium bromide is actually bind in the minor groove. Okay, and by this it stains the DNA band. So agarose gel in casting tray, it is like that. This is the casting tray, and this one is gelatin like is actually is gel. And the supercoiled DNA DNA molecules travel faster as compared to the linear and nicked molecules. Okay, in the supercoiled DNA molecules, it is travel faster as compared to linear and nicked molecules. It is like a principle. And the same sized DNA bands migrate slower in gel with higher percentage of agarose. It is actually about is if the percentage, kindly keep in mind and also write in your notebooks, if the percentage of agarose, if the percentage of agarose is greater, is high, okay, if the percentage of the agarose is high, the size of the pores, the size of the pores are small. And this is the actually the apparatus of gel electrophoresis, in which this is the power slide, this is the caster, and this is the container. And how we visualize the DNA or RNA, as already we use the word Ethereum bromide. And if you don't have Ethereum bromide, you can also use the cyber gold, cyber green. In the real-time PCR, we use cyber green, cyber gold, and we also use gel red and gel green like that. In Ethereum bromide, we got that type of that pinkish color. And then in the cyber gold, you got the golden. The cyber green, you got the green and the gel green and the gel red. And the supercoiled and linearized molecules. And this one are the good plasmid preparation. And these are the overloaded gel. And this one was a poor because there is a like supercoiling and like that, and there is a expansion. So it is the poor preparation. And the plasmid prepared with the genomic DNA and RNA contamination. There is a DNA and RNA contamination like that. And not only a gross gel electrophoresis, we also use the vertical gel electrophoresis and denaturing gradient gel electrophoresis like page polyacrylamide page gel electrophoresis. Polyacrylamide gel electrophoresis. And so if you have any questions and about the quantification like NanoDrop and the gel electrophoresis, you can ask me in the chat box or by using the mic. Okay, Zainab, you have a question. Between nanotroph and gel electrophoresis, which is more advisable to use? I think so. If your extraction is DNA, okay. So first of all, you should go towards the gel, okay. You got both answers. Actually it is not about the quantification, but you can assume what is actually the quantification. of your DNA. But the quality of the DNA is only possible by using the gel electrophoresis. In that, you are not just assume. You got actually what is the quality of your DNA. So I think so. In the NanoDrop and gel electrophoresis, you should go towards the gel electrophoresis if you consider the quantification, the quality of your DNA or RNA. But if you want to just quantify your sample or the DNA or RNA, then you should go towards the NanoDrop. Uh, if you have both facility in your lab, I think so you should go towards the both. First of all, you should go towards the NanoDrop and then towards the gel electrophoresis. So I think so your answer is, your question is answered. Huh? Okay, now we're going towards the PCR, the polymerase chain reaction. Like the polymerase chain reaction, this is actually the sorry, the thermocycline. Okay, what is the principle of polymerase chain reaction? Sorry, what is the principle of PCR? It is a technique used in molecular biology. Okay, in vitro. Okay, in vitro it means in the lab. A DNA polymerase used to amplify the piece of DNA by using enzymatic enzymatic replication. The DNA generated is used as a template for replication. A chain of reactions start in which the DNA template is exponentially amplified. And exponentially means 1, 2, 4, 8, 16, 32, 64, 130, I think so 64, 128, and like that to it amplify even a single piece of DNA to millions or the more copies in just 30 to 35 cycles. Amazing. And it employ a heat stable DNA polymerase, like such as Taq polymerase. And DNA polymerase enzymatically assembles a new DNA strand, like this is DNA. DNA is stacked. So by this, the new DNA strand is formed. Single-stranded DNA used as a template and DNA oligonucleotide. It is uses thermal cycling. Okay, like the thermocyclers. So the PCR, for the PCR, what is the requirements? DNA template. First of all, we use the DNA template. Require having the target region and the two primers. First of all, the forward primer, and the second one is the reverse primer. And the DNA polymerase, such as Taq polymerase, the DNTPs, the buffer solution, the divalent cations. The volume used for PCR is 20 to 150 microliter in small reaction tubes like 0.2 to 0.5 milliliter volumes placed in a thermocycler. The thermocycler heats and cools the reaction tubes. Okay. The thermal cycle we use here is actually heats and cools the reactions. Okay, what are the steps of PCR? The Actually there are three major steps. What First of all, the denaturation. This is actually done at 94 degrees centigrade. Okay, and then the annealing that is done at 54 degrees centigrade, and then the extension that is done at 72 degrees centigrade. It is like that. First of all, like that, 0 degrees centigrade, then 10, 20, 30, 40, 50, and also like that to 100. And that there is the 94. At that place, 94 degrees centigrade, and then it comes to, mm, sorry, at this place. Like that, this is at that place, 54. Like there is the 54. Okay, and then at the 72, and then add to the 4 degrees centigrade for holding. So which are repeated for 30 to 40 cycles. Okay, 30 to 40 cycles are repeated again and again. 30 to 40 cycles are repeated again and again. Okay, okay, okay. Now I'm going to show you a new link. Okay, I send the new link to all of you. Kindly join this in just a 1 minute, because in 3 minutes this session will be ended, because this call will be ended. Kindly join this link. Kindly join the new link. Kindly join the new link. Okay, leave the previous session and join the new link. The call will be ends in a minute, so kindly leave this session. Kindly leave this call and join the new link. Okay, hope so all are present here. I am audible. Can you tell me, am I audible? Thank you. Thank you. Thank you. So I am going to share you this screen to all of you. Okay, we're talking about here. The repeats or the cycles are about to 34 to 40 cycles, and it is an automated cycle. So first of all, it will go through one, two, three, and up to the 35. For example, the 35 cycles, and then it repeats again. One, two, three, four, and then at the 35. So they can heat and cool the reaction mixture in a short time. This is the actually the advantage of. So in the PCR polymerase chain reaction, 30 to 40 cycles in three steps, first of all, and then annealing, 45 seconds at 50.4 degrees centigrade, and then step three extension, 2 minutes, 72 degrees centigrade, and like that. Like that. Okay, now how we detect the PCR product? To check whether the PCR generated the anticipated DNA fragment. What we use? Agarose gel electrophoresis we used is employed for separation of the PCR products. The size of the PCR product is determined by comparison with the DNA ladder. Okay, we use there is a DNA ladder like that. This is the gel and this is the DNA. And after 400, there is a big 500, then one, two, three, four, then again 1000, sorry, 500, then one, two, three, four, then like that. Okay, and we read from here to from bottom to top, which contains the DNA fragments of known size run on the gel alongside the PCR plate. Okay, these are the steps. First of all, we use the vortex mixing, then we put it into the thermocycler, and then we micropipette it and just put into the wells, and then we go the bands, and then we check it into the by using the jar dough. If you have any question, kindly ask me or just off your mic. Anna, if you have question, you can ask me here. Uh, Now the gel electrophoresis, what we got here is the PCR products. First of all, the ladder. This one for like first, two, three, four, and then 500. Then one, two, three, four, then again 500. One, two, three, four, then again 500. So what is the size of the PCR product is like close to PCR. Sorry, 500. So there is the PCR product is 584 base pair. Okay, now we are dive into the primer designing. So first of all, we got the sequence sequence. Okay, first of all, we got the sequence. like the sequence of NM203378 in NCBI GenBank, so that the coding sequence of human myoglobin will be amplified using the PCR reaction. First of all, we got the sequence. Just copy it. Just copy that sequence. And then open primer 3. Okay, just open primer 3 and paste it into the that box. Just check in this one. Left primer like uh, forward primer and the right primer like reverse primer. And don't check it. This. Don't check it. And just click on pick primer. Okay, there are also the information of sequence ID, the number to return, the primer size, the primer TM, the product TM, and just click on pick primers. Okay, done it. And what we got, the primers. This one primers. The length of the primers are 20. It is best. It must be 18 to 25. It is best. And the TM is 60 and 59. The difference must be less than 5 degrees centigrade. So it is also good. The GC content is 50% and 45%. It also must be 40 to 50%. So it is also good. And 3 prime 3.0. Yes, this is the perfect primers. Good. So the resources for general PCR will use the primer 3, the primer 3 plus, the primer Z, the third primer, the vector NT1 and 2010, NCBI probe database for real-time PCR, RT primer DB, primer bank, Q primer depot, PCR QPPD, Pearl primer. Okay, if you have any question about PCR and real-time PCR and primer designing, you can ask me in the chat box. If you have any question about PCR and primer designing, you can ask. You can ask in the chat box. If you have any question about PCR and primer designing, you can ask me in the chat box. Okay, okay. And could you please repeat the last two variables assessing the primer quality after temperature difference? Okay, okay. I'm going to share the screen to all of you and then answer the question. Okay, this one. Okay, there is the TM. Okay, and the temperature is 60 to 59. It must be less than 5 degrees centigrade. Okay, and the GC content. It is actually, what is the GC content? Actually, in the DNA, the four types of nucleotides are present: A, T, G, C. Okay, so the GC content must be high. Why? Because GC, the G and C are by 3 bonds, and A and T are bound by 2 bonds. Okay, so if the GC content is high in our DNA or RNA or in the primer, then it will be a good primer. Okay, because the temperature required to denature that primer must be high. So that is why the GC content must be high, and it must be from 40 to 50%. What does it mean? It means if your primer nucleotides are 100, for example, it is 100. So about 40 to 50 nucleotides must be of either GC, either G or C. And these two one any, it must be close to each other, like 6.0, and the second one is the 4.0. The difference is 2. It must be less than 2. And the segment we got here is that. And it's very close to each other. And the size of the length is almost same. So I think so your question is answered. Thank you. So let's move to the next part of the hour. Okay, now we will talk about the bioinformatics tools. It is actually hands-on. Okay, first of all, right here NCBI. Okay, just NCBI, not NCBI, just NCBI. Then click on first of all National Center for Biotechnology, and then write here some gene, like you write here BRCA1, and search here gene. Okay, search here gene, and there is a BRCA1, and just click on search. Okay, we got, oh sorry, BRCA1. Okay, and just click on first of all the Homo sapiens, like that. Okay, just click on first of all the first one. And here we got the lot of things, but I'm just going to tell you the fast A sequence. Yes, there we got the fast A sequence. Okay, just click on it, fast A. Okay, and we got here what, like that. We got what, the sequence of our BRCA gene. Okay, just click, just copy mm, this one. Okay, just copy it. Okay, and just open the new tab and write here BLAST. Okay, what I write, BLAST, and just click and enter, and just click on BLAST basic local alignment. Oh, the speed is very slow. Again, click here. Alhamdulillah. Click on nucleotide blast. There are four types, but click on nucleotide blast. So, what is blast? Actually, in the NCBI, there is the option of blast because, as we know, in the NCBI, we can write in the search box the gene, the author name, the SNP, the nucleotide. But if you have sequence, okay, like the sequence we copy, like paste it, and we just want to know what which species has this sequence. Okay, for this one, for this purpose, we use BLAST. We have what we have sequence, and we just want to know the species related to this sequence. Okay, and the in the formal one, like in the NCBI, like that, we use like in the NCBI. In this, we just use BRCA1. Okay, this is actually we know the species, the gene, the nucleotide, but we want the sequence. But if you have sequence, you can also know uh, about the species and gene. So just click on blast. Okay, it will take some time. It is just working on your submission. Okay, we got the answer. Okay, it is 100% match to what the BRCA. Hey, congratulations, because it is actually BRCA. So in the alignment, we got the query is and the subject is. Okay, it is 100% matched to what to what to the BRCA. But it is also matched 100% to the musculus strain. So it means that there are the lot of sequence present in one species that is 100% matched to the another species. So by this, we can also relate the different species to each other, and doing this one is called the evolutionary relationships. So on that basis, the whole subject is involved around it, and that subject is genetics and evolution. Like in the taxonomy, you got like that the root, the hominidae, the Homo sapiens, the primates, and in the alignments you got this is 100%. Okay, the e value is zero. The e value must be zero because the if it is randomly matched, okay, it is not randomly matched. It is actually no to match, way to match. So the gaps are 0%, and the scores is very high, and identity is more than 99%. So by using plus, you can do this. I hope so you understand it. If you have any question about blast, you can ask me here. If you have any question about blast tool, you can ask me here. Okay, Zainab, actually, okay, Anna and Zainab both have question. Actually, uh, sometimes we do DNA extraction. Okay, then we just directly going through this sequencing, like Sanger sequencing and next generation sequence. By this, we got the sequence of that DNA. But we don't know which species has this sequence. Okay, so in order to know the sequence, the species or the gene that has this sequence, we have to go through the BLAST tool. Okay, and by this technique, you can also relate the different genes and species can be done. So, uh, I think so. Zainab's question is answered. If you still have question, you can ask me here again. And Anna has question. Could you give an example of disease how it could be applied? Okay. For example, Anna, uh, in your clinic, for example, in your clinic, it is just for it. Uh, some SMA or like hemophilia diseased person is come to your clinic. You just and you by the signs and symptoms you got the answer. The patient has hemophilia. Okay, but you are not assured. You are just assume maybe have some other diseases because by the signs and symptoms you can't actually uh, predict what actually disease it got. So for this you have to go through the sequencing. So first of all, what you do, you just sit down. Let's sit down the patient and take this sample, blood sample, and then you go through the DNA or RNA extraction. Okay, and then you go through the sequencing. And after this, you got a sequence. When you got this sequence, you go through the blast. Okay, you put that sequence. What you got by sequencing, you just paste it into the blast tool, and just click on blast it. And now what you got, it is 99% or 98% similar to the gene that is actually mutated in the patient. Okay, if the patient or if the person is 100% healthy, this sequence will be shown to you. But in the patient, there is a different sorry, there is a point mutation or the just difference of 1 to 2 nucleotides. Okay, and when you compare it to each other, you got nothing. You got that there is a mutation. Okay, for this, I am just going to share you the screen, all of you. Okay, like that. We just open the Word file, and what would go here, just paste it here. Okay, like that. This is actually the uh, sequence of BRCA1, and it is healthy one. And what we just go here. Okay, and I am just going to mutate this one. Okay, we just go to mutate this one. Like we just go to mutate A with just delete A and just put C here. Okay, and I'm just going to change the color. Okay, and we both know, we all know that we mutate the sequence. And after which one? One, two, three, four, five, six, seven, eight, nine, 12. It is like after, for example, after 30 words, we mutate one. Okay, and just Control A and Control C. Just copy that sequence and again use blast. Again blast and just copy paste here. Okay, this sequence is mutate one. Okay, for example, this is the patient's sequence, and just click on blast. Okay, now it's just going to take some time. Okay, now we got the. Okay, now it is 99.99%. First one, with the sequence, when the sequence is 100% same, then there is a there is a 100% percentage entity. But now it is 99.99% because why? Because we mutate the sequence here from there. Okay, we mutate the sequence from there. Like in the alignment, we also can also check that from there as it goes. Okay, like that. It is just tells us there must be a, and we know we replace the A with C. Okay, so they tell us there must be A, and there is the change is C. So by this, the mutation can be same, and by this we know the answer that the patient got that mutation in that gene. So that is why he has breast cancer, colorectal cancer, or a hemophilia like that. Hope so you got the answer. Anna, do you got the answer? Okay. Now we just jump into the mini task. Okay. Just take screenshot of it. The deadline is the before the day 3 session begins April 20, 2025, 9 p.m. Submit on second day. Sorry, on third day. Best submissions will get shout outs during the next session. Task overview. You have learned how to collect samples, design primers, and check DNA quality using NanoDrop. Now it is time to apply it. Okay. So what is the question? Again, take the screenshot of it also. First of all, choose a gene of interest. Okay. Like the BRCA1, the TP50, APC, insulin. Oh, pick any gene you are curious about. Okay. Search the gene on NCBI. Okay. Use NCBI and find the full gene sequence, like the fastest sequence, like in the hands-on practice. Copy a small portion of the coding sequence, around 500 to 800 base pair. And then again, and in the third one, design primers using Primer-BLAST. Okay, use primer like primer 3, primer 3, and paste your sequence and generate a forward and reverse primers. Screenshot or copy the sequence. Kindly screenshot or copy the sequence, primer pair, and product pair. Answer these questions briefly. This is the assignment for the third day. What sample type would you use to isolate this thing? Okay, for example, just assume you got the sample of blood or saliva. It is just assume. Why did you choose this gene? For example, you choose the APC gene. Why you choose this gene? Answer this briefly in the word file. What is the expected product size of your primers? Okay, you got what you got, and the next one, the last one is what is the A260 to A280 ideal range for pure DNA? It is I was just shared to you a 260 by 280 range is 1.8 to 2.0. Okay, so answer the all questions into the word file and kindly submit on the day third of the last day of the workshop. So thank you for being part of this wonderful journey. Follow for further workshop announcements and lot more. The LinkedIn.com Alidras, kindly make connections on and also follow me on LinkedIn kindly. And also kindly, it is Alidras. Kindly subscribe on my YouTube. On YouTube, you will also got the recordings of our sessions and our another workshops. And if you have any query and any question, you can ask me in the email. And if you have any question, you can also contact me on by using WhatsApp. And if you are doing your research, you can also contact me. We will definitely help you out in it. Thank you so much. Okay, okay. Anna, okay. Question in the chat box. Anna, if you have any question, ask me in the chat box. Omid, the workshop is start at 9 p.m. Pakistan Standard Time. If you are from India, it is start almost at 8:30 p.m. Because in India right now the time is 11:05. Okay, at 8:30 p.m. the workshop was begun. Anna, ask me question in the chat box. Okay, if the primers are not good considering the parameters, what steps to do we do? Okay, uh, now in the primer 3, you got almost 10 pairs of your primers. If the first one is not good, go to the second. If the second one is not according to your parameters, go to the third one, the fourth one, the fifth one. In the 10 pairs of your primers, one of it is according to the parameters. It may be a good considering to your parameters. Okay, so you can select. But in the 10, in the all 10, you got not what you actually want to get. So what you do, you can change the sequence because you just copy the first 800 sequences. So then you should go to the, okay, would you mind showing on the web? Okay, okay. I'm going to show you on the web. Okay, like that. Just copy paste it. We just control C and we write here primer 3. Like that, primer 3. And we paste here and click on pick primers. Okay. Now you got here is left primer, that is 5090 and 59 60 55. Okay, it is also considerable. Length is almost same. Okay. Now at the end, okay, it is the first pair, this is the second, this is the third, this is the fourth, and the first one, that one is the fifth one. Okay, the five pairs of the primers you got in the primer three, but in the primer one you got 10 different pairs of the primers. Okay, if you don't like that, the GC content is 55, 55. This is very good. It is 50, 58, 58. It is also very good. But no, it is not good. It is 19.6. So you have to go to the next one. You can go to the this one. The fourth one is very good. 20, 20, 59, 59. It is very close to each other. 55, 55 is very close to each other. It is very close to each other. Yes, it is after 0, 0. It must be 0. Hairpins and 3 prime hairpins must be 0, because if the hairpins created, your primers will be not good, and the length is very close to each other. So you can select that one. Hope so your questions are answered. And your question is answered. Okay, thank you. If you have any question, Omid, Duria Carver, Anna, if you have any question, you can ask me here. Anna, you have two questions. If you have question left right now, you can ask me in the chat box. I will be very glad to answer you. Okay, thank you so much for this wonderful journey. We will definitely join at the third session. Okay, Anna has a question. Do you know, for example, in patients with the dilated gut? Okay, Anna, kindly contact me on this number. I am just sharing you. Kindly contact me on this number. Oh, I think so. Yes. Kindly contact me on this number. We will definitely answer you because it is very personal and it is very specific to you. That is why I share you the number. You can ask me here. I will definitely answer you. Okay, thank you so much for this wonderful journey. We will definitely meet on the third session. Allah Hafiz.